Our next speaker is Professor Marinka Zitnik from Harvard University, which is quite close to MIT, both geographically and in terms of collaborations. So Marinka, I would let you start.
Great. Hello, everyone. Thank you so much, Regina, for this invitation. It's my pleasure to be here today. So today I will talk about graph neural networks for COVID-19 repurposing. So the problem of drug repurposing is something that does not need to be strongly motivated to this audience here. Given the disruptive nature and speed of the pandemic, the traditional methodologies that rely on development and iterative development of drugs are really not feasible these days. And so we need new strategies that can rapidly identify repurposing opportunities and then be quickly deployed in real world. And so the question that we are tackling here is really how can we find these new tricks for drugs that are already in the market or that are in later clinical stages of drug development, and how can we repurpose them in a much more fast manner at the pandemic scale to COVID-19 in particular. And so there are a number of challenges that need to be solved to realize this vision. And the group of challenges I'll be focusing on in this talk is related to understanding the effects of drugs and really leveraging that understanding in order to prioritize drugs for their efficacy in treating COVID-19. So the effect of a drug is not really limited only to molecules to which the drug directly bind in the human body. Instead, the effect of drugs propagate through underlying biological networks. And similarly, diseases are often not independent of each other, but there might be a number of genes that are shared across quite distinct diseases. And so because of that, effect of a drug on a disease is inherently a network phenomenon. So my group was very excited to try to leverage this understanding in order to contribute to the response to the pandemic. So in today's talk, I will outline a journey that we started on March 23rd. The journey is still ongoing. And that journey was really about rapidly deploying our toolbox of AI algorithms in order to identify repurposing opportunities, and then also experimentally screen and verify them and also deploy them in practice. And so all the results that I'll be describing, the methodology, as well as data sets, including experimental readouts and confirmatory studies of predicted drugs, are available online. The preprint of the paper can be found on arXiv. We also have all the data available from our lab website. So one of the main challenges for making predictions related to COVID-19 is that this is really a never before seen disease. And so what that means in the context of machine learning is really that we need to think about how to make predictions in the zero-shot learning setting, how to make predictions for COVID-19, which is a disease for which no good treatments exist yet. At least they have not in the beginning of March when we started working on the project. So at the very high level, we can think of this question as having a large bipartite graph. We have drugs on the left. Those are drugs that are approved on the US market, drugs that are in late stages of clinical trial development. And on the right, we have a large number of diseases. So we want to take many diseases into account when modeling these problems, because we know that there is lots of information that we can borrow and share across diseases. So really in this bipartite graph, edges connecting drugs to diseases indicate known treatment or approval information, also known as indication. The question then becomes, how can we predict what drugs will treat COVID-19? So how can we establish these new links between drugs and COVID-19, given that there are no existing links between any drugs and COVID-19 disease yet? So that was the question that we were tackling. And the first to answer this question, the first step towards that is to think about how can we really represent COVID-19 from computational standpoint. So how can we computationally operationalize it using the networks paradigm? And so for that, we relied on an amazing effort done at UCSF by Gordon et al. And others that have identified around 300 SARS-CoV-2 targets. And so what we did was we mapped SARS-CoV-2 targets onto the human interactome to identify, well, what are the human proteins that are targeted by the viral proteins. And in doing so, what we get a network neighborhood of the human interactome, of the human protein-protein interaction network, to which viral proteins bind, and they are perturbed by SARS-CoV-2. And so that gives rise to a COVID-19 disease module or COVID-19 subgraph of the interactome. So how does that subgraph look like? So COVID-19 subgraph is then defined, or COVID-19 disease module is then defined as a set of human proteins that are targeted by SARS-CoV-2. And on the left part of the slide, I'm showing the largest connected component of the human protein interaction network of COVID-19 disease module. We were looking at what is the structure of that module and whether are genes that are in this module, whether they are expressed in certain tissues that are affected by COVID-19, and indeed that is the case. And we looked at a variety of different tissues using data from GTEx and others. And so here on the right part of the slide, you can see how the largest connected component is of COVID-19 module is then expressed both in lung interactome that consists of lung-specific protein-protein interaction network, as well as full human interaction network. An interesting point here to make is that it is interesting to see that that COVID-19 module is somewhat localized. So it really forms its largest connected component. It's not the case that human proteins targeted by the virus are just randomly distributed throughout the underlying network, but they're really localized in a particular region of PPI network. That's why we see this largest connected component, which is much, much larger than what would be expected in random cases. And so this is really the key insight that we have leveraged when developing the ML models, this insight of subgraphs that allow us to approach the problem of drug repurposing in the following way. So we think of having a large underlying human interaction network, which is shown in this slide in the form of the white nodes and black edges. So nodes represent human proteins, edges indicate a physical protein interaction network. And we overlay that network with a large number of disease and drug subgraphs. Disease subgraphs are defined as subgraphs of the PPI network defined on disease-associated proteins. One of those subgraphs is COVID-19 subgraph that we have just seen. Drug subgraphs are defined by rich protein networks defined by drug targets. And so in pharmacological sciences, there is this common notion that a drug is likely to treat a disease and if it is close to the disease in some pharmacological space. However, that is a very abstract notion, and we were wondering how can we computationally operationalize it. And so the idea was let's use the power of the paradigm of embeddings to measure closeness in the pharmacological space by really measuring closeness between these drug and disease subgraphs and leverage that for prediction. So why are subgraphs challenging to model from ML standpoint? So here really means that we need to move beyond classic graph machine learning prediction tasks that are mostly concerned with predicting properties of individual nodes or predicting links between nodes. We need to do here prediction over structures of varying sizes, because disease and drug subgraphs can be of varying size. They do not necessarily represent some notion of K-hop neighborhoods. And each particular subgraph might actually consist of multiple components that are far away from each other. They have rich connectivity structure, and it's important to take into account the internal structure of disease and drug subgraphs, as well as their border structure and where exactly in the interactome they are positioned. And finally, what is really important is to think about whether those subgraphs are localized within a region of a protein interaction network, as I have shown before for COVID-19 module, or they might be distributed across multiple neighborhoods. So these were some of the challenges that have motivated development of our method that allowed us to take as input a large number of drug disease subgraph pairs shown on the left, and then learn powerful embeddings for those drugs and disease subgraphs, and then leveraging those embedding in order to predict whether a particular drug should be indicated or contraindicated for the disease, and really making those, training the model on known information about approved drug indication, drug disease pairs before applying it to COVID-19. And so the model that allows us to do that is called SubGNN. It stands for Subgraph Neural Networks. And the idea here is really that we specify powerful neural message passing schemes that propagate neural messages from drug and disease subgraphs down to the underlying a graph, which is in our case rich protein interaction network, and then aggregate information from neighbors and neighboring proteins in that interaction network, and then pass those messages back to drug and disease subgraphs. And that in doing so, we really operationalize the concept that the effects of drugs are really not limited only to proteins to which they directly bind, because we can propagate those neural messages along the underlying protein interaction network. And we take into account a variety of interesting subgraph topology that I mentioned before when describing the challenges. So what we did in March was then we run and use those algorithms to learn an embedding space of drugs and diseases, including COVID-19. And then we asked, well, where in that embedding space COVID-19 can be found, and where, or the subgraph of COVID-19, where does it embed, where is it embedded, and what are neighboring drugs and diseases. And so that allowed us to start formulating prioritized lists of repurposing opportunities for COVID-19 by looking at what are closest drugs to COVID-19 embedding in that space. So in order to get some understanding into how good our predictions are when we make them, we first thought of a computational setup and before we proceeded with experimental verification. So our computational setup back in March and April was that we really wanted to first use some computational strategy to get understanding into the potential accuracy of our predictions. And so for that, we used the following proxy for ground truth information. We monitored drugs that were at that point in time in April under clinical trial development, under clinical trial tests for COVID-19. And there were actually a number of clinical trials, and that is really those clinical trials in many ways capture the medical community assessment of drugs. What drugs might potentially work for COVID, not really what drugs actually work for COVID. And so because of that, that was a very weak, perhaps bronze standard proxy that we use for ground truth information to get some understanding in how accurate our predictions are before proceeding with experimental validation. So in April 15, we finalized our predictions. We released those predictions and shared them with biomedical community, as well as we looked at clinical trials in order to see where our predictions stand at that point in time. And so this slide is showing on the left part, the true positive versus false positive rate of a variety of different prediction pipelines that we tried out, where we used clinical drugs under clinical trials development as ground truth information to calculate performance. So we employed a number of algorithms that were developed in the last 20 years or so from the field of network medicine and network biology, including many popular proximity diffusion-based metrics. And what I'm highlighting here on the left part are 4 prediction pipelines that are actually graph neural network-based methods that really have performed outstandingly well, and that has motivated us that while all predictive pipelines have some predictive signals, graph neural networks are particularly strong predictors, and that has then motivated us really to push forward those predictions to experimental validation. So in April 15, we generated this ranked list of repurposing candidates. Here I'm just showing a top list of drugs that were predicted by our final algorithm that we call C-rank, that has prioritized and aggregated a number of different ranked lists. And we were then wondering really, well, how can we take that list and really move beyond just computational in silico prediction of our algorithms? How can we show that they are really that they have strong signal beyond just assessment in clinical trial data? And so for that, we teamed up with National Emerging Infectious Disease Laboratories at BU, and our colleagues at BU Needle were excited to experimentally validate our predictions. So they took 918 compounds that constituted a top part of our prediction lists, and they screened those compounds for efficacy against SARS CoV2 in vivo E6 cells. So these are cells that are epithelial kidney cells of African green monkey. And out of 918 compounds that they have experimentally screened, 800 had no detectable effect on viral infectivity, 35 were cytotoxic to host cells. So that means that really those are drugs that would kill human cells, so they are not good candidates. 37 drugs had strong effect, being inhibiting viral growth and keeping human cells alive, and they were active over a broad range of concentrations and drug dosages, and 40 had weak effect on the virus. And so by because our collaborators were willing to generate this large screen, then we were able to really go back again and say, well, given now these ground truth gold standard labels, because these are really about how these 918 drugs work for COVID-19, where do we find these drugs in our predicted lists? Are really predicted close to the top, and is there anything common to those drugs? So one of our key findings was that if we look at strong effect drugs and weak effect drugs, so those drugs that have inhibited viral growth and at the same time ensure that human host cells were alive, 76 out of 77 drugs that we have successfully predicted and that also have been experimentally validated, we realized that they do not bind to proteins that directly bind to proteins that are targeted by the virus. So what does that mean? That means that these drugs really rely on network-based actions. We could not find those drugs if we would be using more classic drug repurposing strategies based on, for example, docking methodology that would simply prioritize drugs based on the direct sharing of drug targets and viral host proteins targeted by the virus. And so we call this a set of drugs, we call them network drugs that really rely on the notion of network effects in order to make to be effective for COVID-19. So going to this exercise between March actually, and June, end of June, beginning of July, where we have rapidly deployed our ML algorithms and then really also see how they can inform and guide experimental validations and successfully validate predictions. There are a number of machine learning lessons that that I have learned about. And so I would like to just share two important lessons here with you. So first is that domain scientists, in our case, those were biologists, virologists, and medical doctors, we really found out that they need to have mechanisms. We need to give them mechanisms to interact with AI systems so that they can get understanding into what are the part of the data that ML algorithms are making decisions based on, and those domain experts also need to be able to provide feedback for the machine learning loop that would need to be integrated back in the model. So it's really, it's not sustainable to be simply sharing and sending CSV tables back and forth between computer scientists and biologists and virologists. We are really now thinking and developing these systems that not only generate predictions, but also provide explanations that turned out to be very useful for design of downstream experiments. And second point, is that generalizing to new diseases, generalizing to diseases that were never seen before is extremely challenging. And so many drug repurposing opportunities exist in the literature. If you read papers, you might find that many papers report extremely high performance metrics and high AUC scores, high precision recall scores on a variety of benchmark data sets. However, most of that accuracy does not really translate to real world cases when these models are tasked with making new predictions. And the primary reason for that is that many of these algorithms often rely on the fact that there is already a particular drug that exists for a given disease. And so they are doing simply guilty by association type of predictions, which of course completely fails when we are tasked with new diseases. And those are really uninteresting, important, challenging questions where AI can help a lot. Okay, so I will stop here.
Marinka, thank you very much.
Yes.
I didn't want to interrupt. We're really running out of time now. So Marinka, thank you for an excellent talk, a lot of thought-provoking questions.